Good day to you all. This short talk is on muscle fibres and their significance. I'm indebted to Professor Tacken for his assistance with this. You may pause this presentation at any time in order to study a slide. A muscle is made up of many muscle fibres, and a muscle fibre is made up of many myofibrils. It is the myofibril that contains the contractile unit called the sarcomere. And the sarcomere are short little areas separated by a Z disc. So there will be a sarcomere here and a sarcomere here. But this is the contractile element of the muscle. There is no individual innervation to a myofibril. The only innervation is to the fiber, which is done via an indentation of the sarcolemma. If we look at this schematic, this shows a thin filament of actin attached to the Z disc we referred to. And this then is the myosin, which is much thicker filaments. And that has a constant width. When a contraction occurs, this doesn't shorten in any way. As you will see, it is the myosin drawing the actin into the tube that produces the contraction by bringing the Z disc closer together. Well, how does it achieve this? Well, this is your actin thin filament, and this is your myosin. And the myosin has a head on it of many hinged areas. And when that forms together like that, they're called a cross bridge. The myosin then contracts, they'll put it this way, contracts and pulls the actin filament into the A band. And this is the power stroke and requires hydrolysis of ATP to release high energy phosphate. If we now look at this actin filament, this is the actin filament here, you'll see that there's other tiny filaments running through it. These are tropomyosin, and the tropomyosin filament is actually covering the myosin binding sites. So this means the myosin can't do anything to the actin. But with presence of calcium, troponin, not tropomyosin, troponin will move the tropomyosin to uncover the myosin binding sites. Now the myosin can bind to the actin filament and move it in to the A band. The calcium is derived from subsequent neuromuscular junction depolarization, which then diffuses into the myofibrils. In muscles where fine control is required, for example, eyes, only a few dozen fibers may be supplied, but in large muscles, the gluteus, precision is not so important as many thousand are supplied. The motor neuron with its accompanying vascular and lymphatic bundle are termed the motor neuron unit. The arterial component has smooth muscle up to the terminal arterioles. The lymphatics have no smooth muscle and rely on the muscle contraction to clear fluid from the muscle. The smooth muscle gives some control over the blood flow in the vessel coming down, but once it gets to the terminal arterioles, it can't exert any control. The mechanical efficiency of skeletal muscle is low, probably as low as 20%, with a range of somewhere 18 to 30%. The fast fibers are less efficient than slow fibers. Only a proportion, though, of the ATP is used for contraction. 75% is used for heat production. During normal activities, core body temperature is maintained by thermogenesis of skeletal muscle, and to a lesser extent by certain adipose tissues. Shivering is, of course, an extreme example of contractile thermogenesis, but not non-contractile thermogenesis by muscle is achieved by complex metabolic changes, often termed futile, as they appear to serve no other purpose than to generate heat. It appears, therefore, that the muscle is an organ that is dedicated also to maintaining body core temperature. It's also of interest to the author that under anesthesia associated with neuromuscular blockade, there is apparently no non-contractile thermogenesis, as the maintenance of body core temperature can be a challenge during extended surgery. Before we move on to a description of the various fiber types, it is important to know that controversy exists over the typing, and I'll refer you now to this article. That article is very high-powered, but nevertheless you can take away some important messages from it. The classic view of skeletal muscle is that there's a heterogeneity of motor neurons defining the typology of the fibers within a motor unit, with fiber types type 1, type 2A, and type 2X in humans. However, skeletal muscle fibers are increasingly being viewed as not being discrete, but along a continuum, fueled by identification and acceptance of the existence of hybrid fibers. So, in this article, extensive analysis and high-powered analysis techniques, they cannot distinguish between type 2A, type 2X, or the hybrid 2A, 2X fibers. Having said that, let's look at the more usually quoted fiber classification. 3 types of muscle fibers are described. Type 1, slow oxidative, slow twitch, fast oxidative, fast twitch, glycolytic. 2X, not oxidative. They may have a similar structure, but have a very different metabolism and function. The type 1 fibers have a very high density of mitochondria and are thus able to generate ATP via aerobic phosphorylation. They have a small diameter and can only produce limited tension, but have a better resistance to fatigue than any other fiber. Type 2A fibers are similar to type 1 in that they can generate ATP aerobically but contain less mitochondria. They are less resistant to fatigue than type 1. Type 2X, these fibers do not require oxygen. They are able to store energy that can be utilized for short bursts of activity. The aerobic metabolism finally produces lactate, which then goes on to produce muscle fatigue. Pardon me. To a lesser or greater degree, muscle fibers are able to store oxygen due to the presence of myoglobin. The concentration is higher in type 1 fibers than type 2. Myoglobin is found in not only muscle cells, but also in the high concentrations in the heart. It stores oxygen by virtue of a radically different association curve to hemoglobin. Myoglobin facilitates the diffusion of oxygen from the extracellular space to the mitochondria. There's only enough oxygen stored for a few minutes of muscle activity. Out of interest, this storing of oxygen is of paramount importance to animals that dive. This then is the dissociation curve of myoglobin. You can see it's dramatically shifted to the left. You can see that at a partial pressure of 10, myoglobin is still 70% saturated. But as it falls, large amounts of oxygen are dramatically released. That is the way it's acting as a store for oxygen. For more detailed information on this, you will obtain this from its reference. But what does all this mean, and what are the implications? Fast switch fibers contract faster than slow switch, as they are able to break down ATP twice as fast as the slow twitch fiber, because they have double the amount of myosin ATPase. The oxidative slow twitch fibers fatigue at a slower rate. This makes them important for long-lasting activities such as running or swimming. On the other hand, the fast twitch fibers can reach intense bursts of activity such as sprinting or weightlifting. This has very important considerations. Everybody has all 3 types of muscle fiber, but the ratio one to another varies considerably from person to person and is also affected by various training techniques. Long-distance runners will have a preponderance of slow-switch fibres, at least 7 to 80%, whereas weightlifters will have a dominance of fast-switch fibres, around 60%. One marathon runner in particular had a preponderance of slow fibres of 90%. Because of these variations, a sprinter is unable to run any real distance, and a long-distance runner is unable to sprint, all this being dependent on which type of muscle fiber predominates in that individual. Considerable controversy exists as to the extent of the change in muscle fibers that can be produced by specific training methods, and is beyond the scope of this video. However, if you're interested in that, I'd refer you to that reference. Thank you all very much.